from Microbe TV, this is TWiP. This Week in Parasitism, episode 146, recorded on January 30th, 2018. Hi, everybody. I'm Vincent Racaniello, and joining me today here in a very cold and dark New York City, Dickson Despommier.
Hello, Vincent.
And from a remote location, Daniel Griffin.
Hello, everybody.
Hey, Daniel.
Welcome back, everyone, for another episode in Parasites.
Indeed.
So let's get right down to business, because you guys are seemingly serious as anything.
We're frothing at the mouth.
You don't want to...
Which is case number 47, but no, that's not it.
Daniel, remind us of what was going on.
I am ready for business. So let me remind
the doctor is in.
Let me remind everyone who's clicking back in and acquaint all those who are new. This was a briefcase. We presented a case of a
briefcase. I thought this was a patient. I'm sorry.
It's true. Okay. This was an individual in his 40s. It was a male visiting New York City on business. Originally is from Thailand. And when he comes to New York, he's having some problems with diarrhea and abdominal pain. He reports that the diarrhea is continuous. It's occurring even at night. His belly feels swollen. He feels swollen, actually, is the way he words it. His stool cultures come back negative, but his complete blood count reveals eosinophilia, and the clinician orders a stool, ova, and parasite exam. And as we said last time, something was seen in the stool. We did not use the word egg. I don't believe we used that, no.
We did not.
We said large elliptical objects over 100 microns in size are seen in the stool. And then we left people with that.
All right, we had 6 guesses.
All right.
Number 1, Elizabeth writes, a parasite. I would need to see it. Amoeba or toxoplasma? That's my guess. Wow. Dickson.
David writes, Dear hosts, I would like to take a crack at the case for episode 145, but this one was certainly tricky. The man from Thailand suffering from continuous diarrhea and abdominal pain may be suffering from a case of heavy fasciolitis caused by the liver fluke Fasciolopsis busci. Acute fasciolitis marked by eosinophilia, abdominal pain, and diarrhea, which may set in 1 to 2 months after consumption of raw infected water plants, which we now know from past episodes are a common food item in Thai dishes. Assuming the man's symptoms didn't set in until after he arrived in America, we can also assume the elliptical objects found in his stool were the eggs of the parasite, which are typically more than 100 microns in length. If Fasciolopsis was not the culprit, I suspect another fluke, perhaps Paragonomus or Kinostoma, or any other parasites with eggs of this size, may be the causative agent. Clonorchus sinensis, another common parasite in Thailand, was ruled out of the running since the eggs of this parasite are too small to be consistent with the objects found in the man's stool. And while continuous diarrhea is a symptom that many parasitic protozoans can cause apart from Bacillium coli, well, they are all much smaller than 100 micron objects found in the man's stool, unless he had multiple infections and the eggs are showing only as a result of the diarrhea stemmed from another source. Taking all of these things into consideration, I will stick with my gut and say that Fasciolopsis busci is the culprit. Thank you once again for the informative and educational podcast. Sincerely, David.
Daniel.
Yeah, I thought that was pretty interesting, that the person, the thought process here. So we'll go through this later, and I think
yes, we will.
Bacillium coli gets brought up, and that actually those organisms can be greater than 100 microns. So it could be an interesting question. What we're seeing in the stool? Are we seeing organisms? Are we seeing eggs? Well, soon we will find out. So Alistar writes, Dear hosts, this is my first time writing into the show. I recently introduced the podcast to my lab colleagues, and we are all excited to regularly discuss these case studies.
Cool.
With the information given to the case study involving the patient from Thailand, the diarrhea and abdominal pain points to an intestinal pathogen. Since the stool cultures were negative and the patient had eosinophilia, my first, and this is in quotes, gut feeling was that this could be some kind of helminth parasite rather than a protozoan parasite. Indeed, the O&P, or ova and parasite exam, implies that parasite eggs were found in the patient's stool. My next step was to consult the CDC website to compare different helminth ova sizes and morphologies. And he did. What about the free downloadable book? Anyway, so we have a link here to the CDC,
Right.
And it's a diagnostic procedures. Let me continue. Since the ova were found to be larger than 100 microns, this narrowed the possible candidates. Schistosome eggs can be this large, but generally do not fit the elliptical shape description, and schistosomiasis, as far as I know, is not particularly prevalent in Thailand. This led me to Fasciolopsis species, and more specifically liver flukes, which use snails as an intermediate host and use many mammals, including humans, as the definitive host. There was a little bit of a grumble going on from one of the TWiP people there. Infection can occur by ingestion of cysts attached to vegetation, such as watercress. Liver flukes can cause diarrhea and abdominal pain in cases of heavy infection. Indeed, there was recently a report of a German traveler to Thailand that became infected with F. hepatica and had all the described symptoms, including abdominal pain, persistent diarrhea, and hypereosinophilia. We get a link there to a PubMed article. Another possibility would be Fasciolopsis busci, which can cause similar symptoms and has a history of being endemic in Thailand. Fasciolopsis busci and Fasciolopsis hepatica have apparently indistinguishable ova during stool examinations. I have to say that if I do win the book, it wasn't because of a mere fluke.
Oof.
Best wishes. Alistair from the University of Manchester.
Manchester.
Mm-hmm.
Eric writes, Dear Professors Racaniello, Despommier, and Griffin, your podcast and its kin have been entertaining and inspiring me on my daily commute since I discovered them 6 months ago. Thank you very much for putting the shows together. It's my first time writing in. My guess is Fasciolopsis busci, the giant intestinal fluke. The chief complaint, diarrhea persisting for what sounds like a week at least, eosinophilia, objects in the stool, and the lack, I assume, of other symptoms and lab tests. Frankly, bloody stools, pain, liver symptoms, labs, bacteria upon culture suggests a helminth infection of the intestine specifically. Unicellular eukaryotes are in the tens of microns long, and 100 microns would be on the large end of the spectrum. For example, I learned from your book that Giardia species and Entamoeba histolytica cysts are both about 15 microns long each. Of the nematodes, cestodes, and trematodes, I only found the trematodes to have sufficiently large eggs. The most common and largest intestinal nematode, Ascaris, for example, has eggs around 50 to 70 microns in length only. So he had an asterisk on largest, and that means I wonder how well the size of an adult worm corresponds to the size of the eggs. Can I argue that the largest worm of a clade has the largest eggs as well? Such a rule seems to work with vertebrates. Fasciolopsis is the largest intestinal fluke in man. Of the trematodes, Schistosoma species and Fasciola species have large eggs, but the patient's clinical picture lacks important symptoms, liver involvement that we'd expect for those infections. Fasciolopsis busci matches my expectations. It produces large eggs around 130 to 150 microns in size that travel in the stool. It is indeed endemic in Thailand and is a common infection worldwide. Asterisk again. A study around 35 years ago found 10% prevalence in a school in central Thailand. A library book mentioned Norman Stoll's 1947 estimate of 10 million cases worldwide, but Stoll didn't cite a source. It was hard to find a more recent estimate for these neglected diseases. Transmission by eating Metacercariae on aquatic plants is plausible for our patient. The worm attaches to the small intestine epithelium, causes diarrhea. In heavy infections, the diarrhea can be continuous with nausea, vomiting, among other complications. There are other species of intestinal flukes, echinostoma species, in humans which produce over 100 micron eggs, many reported from eastern and southeastern Asia, but none as common as Fasciolopsis. Another asterisk. And I've already told you that one. Same one as before. Treatment, praziquantel, at least 15 milligrams per kg. WHO recommends one dose of 25 mg per kg for all intestinal flukes. Gives a link to that. Sincerely, Eric in Los Angeles, California. And then he adds, P.S., I think there's a typo in the table of contents of the book on page Roman numeral 8. Manzanella ozardi is spelled Manzanella with a Z instead of an S.
No, I'm going to check this right now. This we appreciate that.
Not a big deal, but just pointing it out for any future editions.
Thank you.
Dickson.
Yes, Carl writes, Dear TWIPniks, in trying to solve this case, I looked through the egg pictures in Appendix C of Parasitic Diseases, 6th edition. I figured that most parasite eggs are smaller than 100 microns, so that was a good place to start. I found several species with appropriate eggs, which I used to build an elaborate spreadsheet indexing organisms against symptoms. If I were a real parasitologist, I could do this in my head. I was then able to estimate all but one species. Here's how
Eliminate.
Here's how I did eliminate them. Schistosoma is not found in Thailand, except Schistosoma mekongai, whose eggs are too small. Paragonimus westermanni does not cause swelling and causes epigastric pain rather than abdominal pain. I'm assuming that the case report makes this distinction, but maybe epigastric pain is a kind of abdominal pain. Also, Paragonimus would be expected to show some lung symptoms in its acute phase. Fasciola hepatica doesn't cause diarrhea. Fasciolopsis doesn't cause swelling. The sole species left standing is Echinostoma, which is actually a genus that nobody seems to distinguish among. Different species have different size eggs, but some are the right size, and they all cause violent watery diarrhea, eosinophilia, abdominal pain, and edema, hence swelling. And they're endemic to Thailand. So my guess is that the unfortunate traveler had echinostomiasis. I hope he got better, and I hope I win a book. Dickson said that azenic was Greek for known life, but he's thinking of notobiotic. I'm sure he's right. For notos, also bios life. Azenic is without strangers from A without and xenos, strange.
Would you say xenos or shenos?
I would say xenos.
You're going to say azenic.
No, azenic.
Azenic.
That's the way I say it.
Xenos.
Yeah.
Zenic and
He's
xenos.
absolutely right, by the way. I did misspeak. I misspoke.
Thus spake
Zarathustra.
Dickson.
That's right.
Dickson.
In reading
Thus
about
misspake.
I'm going to make that the title of this episode.
Gentlemen.
Thus spake.
Thus misspake Despommier. In reading about Paragonimus westermanni, I noticed that it is usually triploid, but it also is sometimes diploid. And I found a paper describing relatively rare tetraploid forms. How the heck does meiosis work in a triploid species? Is this something that's stable on an evolutionary timescale, or is it just a weird phase that's happening in a few generations before the species settles back into normal, even-numbered ploidy? And how did it get started in the first place? I'm a mere electrical engineer, so I can't work out what's going on from the papers I can Google. I'm hoping you can explain it to me.
Could you explain triploidy?
I know what it is, but I can't explain it. I just know that it's 3 sets of chromosomes. And a lot of organisms are triploid, by the way, mostly plants, and it's particularly true for orchids. So, what that means in terms of their genetic success, their evolutionary success, I should say, is kind of questionable because most organisms out there that are eukaryotic are diploid. Would you agree with that? Anybody?
Well, some plants are triploid.
Yeah, no, I just mentioned one. Some others too. I think corn varieties have some triploidy associated.
Triploid organisms are normally sterile as their lack of homologous chromosomes prevents pairing during meiosis.
Look at that.
So, no worries, right?
I guess, but so they're an anomaly that arises by some kind of a freaky mistake.
Now, Daniel, you know about triploidy in humans, a rare...
You know, it's interesting. You know, when you brought that up, I started thinking, what defines us, right? And so, not all human cells are diploid, right? So you start looking at liver cells, you start looking at megakaryocytes, to get up to 64N.
Really?
So, right. These are things I don't think people necessarily think about. So yes, some cells, like sort of the basic building blocks maybe of the human in development, but once we differentiate, not all our cells are diploid anymore.
But I think of germline cells as being haploid. Right?
Yeah. Yeah.
So that's true, but that's a special case because you want to unite them to make a diploid organism. But do you know of any other ploidy cells in the body besides megakaryocytes? Because those are kind of weird cells, right?
Well, liver cells also will
Really?
have polyploidy as well.
All of them, or just a few?
That's an interesting question. Like, what is the ratio of like different liver cells? I am not sure. Like, what percent are?
Okay.
But no, as you bring up, you think of, it makes me think of the plant world when I start hearing about different forms of polyploidy.
Exactly. And what usefulness that is to them is beyond me, except that one of the most beautiful plants in the world is an orchid. And I was told they were triploid. No, I'm just repeating something I heard because I haven't actually looked. But they have a very large genome. That part I do know. So,
Daniel, you have what? You want to hit another thing?
No. We have one more thing. We have one more thing to read.
So, according to this, the triploid orchids are sterile.
Right.
They don't make seeds.
Okay. All right.
And they can also be pentaploid.
Pentaploid. Wow.
HeLa cells are aneuploid. Every cell has a slightly different number. It's not diploid by any means, but they're all different.
Why don't they call that heteroploid?
Well, what does aneuploid mean?
I think without.
Aneuploid is abnormal numbers of chromosomes in a cell.
Oh.
So you would say that heteroploid if they're all different. Yeah, I guess you could.
I don't know.
Yeah, that's actually the word they use to refer to liver cells. They say they're aneuploid, and then they talk about diploid, tetraploid, octoploid, etc.
Wow.
So,
how about unemployed?
Yes.
Are you haploid, Dickson?
Most postdocs are unemployed.
Dickson, do you remember when you were haploid?
I have a vague recollection of the uniting of the sperm and the egg.
You can't.
No, it's not. It's not something I'm consciously aware of.
Dickson, Daniel, can you take our last guess?
Right.
Yes. 6.
Please.
Chris writes, "Good morning to the 3 wise TWIPers. My name is Chris, and now that I've finally caught up on every TWIP episode, now working on TWIB and TWIM,
Wow.
I can finally contribute and send in my case study. Due to the fact that the man from Thailand had eosinophilia and visible eggs in the stool, I instantly thought helminth parasite. So my first guess was Clinorchus sinensis, due to the location of the patient and the fact that he has eosinophilia and diarrhea, which are possible symptoms of this parasite. But upon looking up the size of the eggs of this parasite, it cannot be. Clinorchus sinensis has fairly small eggs, which was one of the main clues Dan provided. After looking through parasitic diseases, I found Fasciolopsis buski. I believe the man from the case study has Fasciolopsis buski because this parasite can cause continuous diarrhea, can cause eosinophilia, even in light infections, as this parasite likes to feed on columnar epithelial cells, and it commonly causes abdominal pain. The last key that helped me reach this conclusion was that I found a nice picture of one of the Fasciolopsis buski eggs, which is both elliptical and is measuring at 140 by 80 microns in Parasitic Diseases 6th edition. I'm feeling pretty good for my first right-in answer for a clinical case, so I am very much looking forward to the next episode. In addition to listening to TWIP today, I read this cool paper in the Journal of Parasitology this morning, and I think it would be cool here if you could talk about it. I think it talks about some cool life cycle stuff and some good fundamental parasitology. Additionally, it talks about a parasite that was once a case study answer, the raccoon roundworm, Baliascaris procyanus. Once again, thanks for all your work, and I'm hoping to win the textbook.
But it sounds like you already have the textbook.
But I think, as we talked about, right, the actual physical hard copy, there's something more wonderful about that than the electronic version.
Yeah.
So, Chris, P.S., I just finished People, Parasites, and Plowshares, and it was a fantastic read. Mm-hmm. I highly encourage every TWIP listener to go out and get their own copy. You won't regret it. Mm-hmm. And then he gives a link to the paper.
Chris, the check is in the mail.
Do you remember you read some of those chapters on TWIP?
I do.
A long time ago.
I do remember.
I got a chance to snooze while you were reading.
Indeed you did. Indeed you did.
When do I snooze when you're doing something?
I have no idea.
You were doing something recently, and I was falling asleep.
I think I was doing one of the online courses presentations.
Oh, that's right. When I was filming you, I was nodding off. All right, six guesses. We had a lot of Booskies. I love that name, Booski. We had some Paragonimus. We had one Balantidium, right?
We did.
What are you going to say, Daniel?
I was two things I was going to say. Oh, we also had an echinostoma.
Yes.
But I was going to say, so, every so often I get communications from TWIP listeners, saying, hey, you said this and then you did this. And one of the communications that I got this last week was when you announced the free book giveaway, you said you were going to just give it to people that emailed in. And then the first time you give one of you say, but only to those who got it right. And this person felt a little bit put off. They felt like the rules had changed.
Yeah, we changed the rules. Yeah.
Yeah. Well,
So does that disencourage people from sending in guesses?
I actually thought it was interesting. I said, do we want only people who feel confident sending stuff in, or do we want to encourage people to think it through, work it through, and send?
I would say the people who got it right don't need the book.
So only if you get it totally.
If you get it wrong, then we will send you 2 copies.
Well, it's your book, so you got to use this. I would say give it to everyone who sends in a
Just
guess.
give it to one of the guesses, no matter which one is correct, and it's a lottery. Basically, it's a lottery.
Okay,
for participation.
I think we'll make this person happy again.
All right,
maybe they'll keep listening. You never know.
Well,
however,
they'll never find out now that they're stopped listening.
Daniel,
exactly.
we need to cut to the chase here as to what it is, because I instantly guessed one thing, and I made the letters in the air to Vincent as you were talking, thinking I had nailed this, but in retrospect, I may not have. So I'm with that first guess, and I need to see the parasite in order to know what it is.
Well, let's go through our the emails, because I really enjoyed these this time. Not that I don't enjoy them all the time, but I like the way people think it through.
Yeah.
I think that's maybe one of the parts of medicine I enjoy, this sort of thinking it through, putting it together as a mystery, and what clues do we have? Because what's the once you get the report from the parasitology lab, you're like, okay, that's what it is. And then you feel like your Monday night quarterback. You say, well, obviously this, that. So I was actually earlier today kind of thinking this through, like how could a person approach this? And one of the first things, and this is, I guess, addressing several of the emailers, is what we're seeing in the stool. Are these organisms or are these eggs? Mm. And so that I thought was that's one way of approaching it. And so when we talk about something being larger than 100 microns, I like the person who brought up Balantidium coli, because there's not a lot of organisms that you're going to see in the stool that are larger than 100 microns.
Correct.
I think now Balantidium coli, 140, 150, right? It's right in the range that's right. We're sort of suggesting. And, when I give these cases and I give a certain amount of clues, I'm trying to make it possible to some degree. I don't think you can always know exactly. So that was one thing. So are we seeing something like Balantidium coli, or are we seeing the eggs of something else? The morphology, maybe that's somewhat helpful if we think about Balantidium coli is going to be round. And what we're describing is something that's ovoid. So maybe that's helpful in some degree. I try to be honest. I'm not gonna tell you, and then we realized it was sort of a slightly smashed Balantidium coli. So hopefully at some point people are starting to think of eggs that are greater than 100 microns. And then I thought, okay, this is pretty straightforward at this point. But I have to say I was really impressed that someone brought up echinostoma, because you go to the back of our book, and I think we actually got an email. I would love if we have listeners who could tell me if they're still doing this, but we actually got a request from the London School of Tropical Medicine to use page 543, our parasite identification charts at the London School of Tropical Medicine.
That's a high compliment.
Yeah, so I thought that was, and if you go through there, you actually start seeing different eggs of different sizes. And some of the eggs can get pretty big. The schistosoma eggs can get pretty big, right? When you get into hematobium, Mansoni, but then who are kind of the biggest eggs? Then you get into the different, fasciola hepatica, fasciolapsus bouskai. So I was thinking maybe that was helping people a little bit. The clinical, we didn't give a lot on the clinical, except for, well, a couple of things. One was the continuous diarrhea. And that's interesting. Most people usually have diarrhea through the day and then they go to bed. People who tend to have diarrhea that wakes them up through the night often have a small bowel diarrhea. So I was hoping that would give people a little bit of a hint as well. So now that I've given all these hints and thought, Dickson, are you starting to feel a little bit better about?
Well, I want to see a picture.
Yeah, you can't. You have a size. That's good enough.
No, the size fits well. But the ovoid versus round is not always the case with Plasmodium, but that doesn't matter. I mean, you definitely, the first guess was the right guess, but unfortunately she didn't go through the diagnostic tests and the symptoms. I would have liked to have seen a more, everyone could have said, oh, I need to see the parasite before I make a guess. That's not fair. So
yeah, you need to.
So, but now we do need to see the parasite because we're going to have a reveal, right?
Well, you don't get to see it, but I will tell you what it was.
Right. Okay.
I'll tell you what the report came back at. So the report comes back, elliptoid operculated eggs.
That nailed. That would have caused me to tell us something completely
Really?
different. Yeah, absolutely. Operculate egg. Sure, absolutely. That's a feature that doesn't correspond to Plasmodium coli at all.
Yeah. Now we know they're eggs. Now
you know they're, and it's probably a fluke of some sort.
Fasciola type eggs.
Well, it has an operculum, but almost all of the
trematodes,
fasciolaceous-like organisms have an operculum. So that's the way the ciliated stage, the mycelium, gets out of the egg. So it pops open this little lid and swims off into the environment.
They're the right size too, right?
And they're the right size, but there's a whole family of these flukes that fall into this category in that area of the world. So it would be very interesting to see what the description is as we continue the story.
Yes. So they continued. And actually, this I think is important. They report is consistent with fasciolid eggs and belonging to either Fasciolapis busci or Fasciola hepatica group.
Exactly.
And I think that's important. You can't necessarily tell without any sort of molecular testing, just the morphology of looking at these. You might have certain ideas from our clinical case, which one it's more consistent with.
However,
yes.
No, but you could also take a more careful case history, right? You could ask this guy what he likes to eat and where he eats and that sort of thing. And that could rule in and out either one of these two things.
Yeah, I think that could help.
Okay, what did you do with this gentleman?
Exactly.
Well, I think we saw another thing. He got his single dose single day of praziquantel. He actually got 3 doses that day, and then he went along, and the symptoms rapidly resolved.
Great.
So good outcome.
Terrific.
Yeah.
And
but I have to say, the person who put echinoderma, right? You look at that, and you say, those eggs, they're about the right size, right?
Correct.
So,
and they're hard to describe. They're almost nondescript. They defy a few words that would say, oh yeah, that's that egg. You could do that with Clonorchus. Or some of the other smaller flukes, because they have these little lips on the edge near the operculum and they've got a flask shape. But these eggs they're ovoid. What more can you say about them? It's even hard to see the operculum. You have to look really close to see it. So,
I think if I had given that little bit more, but I think that would have taken something away from the case. If I said, there was a conspicuous operculum. Then the person with echinoderma would have said, that,
Right. But my initial guess was Plasmodium.
Oh, okay.
Mm-hmm.
All right.
So 4 people out of 6 got Fasciolapis
They did.
busci.
That's excellent. But we're going to put all of them into the pool.
All of them in the pot. Let's pick a winner.
Yep.
Random number generator. Are you ready? 1 through 6. Give me a random number. Boom. Number 6.
Number 6 is the winner.
That's Chris.
Excellent.
Who loves people, parasites, and plowshares. So
Fantastic.
Chris, please send your address to TWiP at microbe.tv. And if you are outside of the U.S., I will also need a phone number for the shipping.
And remember, these are signed by the
These
authors.
are signed by Dickson and Daniel.
That's right.
And someday they will be worth their weight in paper.
They would be worth as much as Chris paid for it.
Now, are you sure this wasn't rigged, Dickson? People just have to write in and say all kinds of wonderful things about you, and then
No, I did.
coincidentally.
I can show you the page capture from the random number generator.
I'm going to confess and say Chris is my nom de plume, but that's a different story. We won't go there.
Okay. So, I thought maybe we should speak a little bit more about, this particular organism. Should we
Sure.
give people our little, what is it, 60 seconds? Dixon, do you want to?
Right.
And I'll throw in the clinical part.
So wait a minute. Which one do you want me to pick, fasciola or fasciolopsis?
Well, let's do fasciolopsis. Let's do blue sky. I think that's
Blue sky.
Now, as I recall,
Blue
this comes
sky.
the parasite lives, as almost all flukes live, with an aquatic stage. The parasite lives in the gut tract, and it feeds on columnar epithelium. It's very large. It's the size of a small leaf. If you connect your index finger with your thumb and look at the hole that results from that, that's about the size of the organism. So they're big. They're very large. They produce eggs. They're hermaphroditic. They self-fertilize for the most part, but they can cross-fertilize also. The eggs are passed in the stool as unembryonated eggs. They have to be deposited in fresh water in order for the cycle to continue. The eggs then embryonate. That is to say, they undergo embryogenesis in the water, producing a miracidium. The miracidium is a ciliated, free-living stage of this parasite, which then hatches from the egg. And I think it's stimulated to hatch by exposure to sun. So the sun shines on the eggs during the morning of its full development and causes the organism to release enzymes, which then digests the seal that the operculum makes with the eggshell itself. Out pops the miracidium, swims around, finds a snail. The snail then becomes infected. The snail then gives off Siccaria, just like the schistosomes give off Siccaria. All the trematodes do this. That is the ones that infect humans. And then the Siccaria go swimming around, and they look for inanimate objects along the shoreline of the lake or the pond that they're dropped into. And I think that Fasciola hepatica mostly insists on plants of the blocking on the name, watercress.
Watercress for hepatica, but similar. I think they're both
the water chestnut,
littoral.
Yeah, but water chestnuts are grown in farms. They're raised in these ponds for consumption at the commercial level, and those are perfect places for these parasites to gain a toehold. And people who work in these areas usually don't have access to sanitary facilities, and so you have a way of getting the eggs into the environment. You've got snails living in these ponds that the water chestnuts are being grown in, and then there are people that actually sit, and after harvesting the water chestnuts, they actually place the water chestnut in their mouth, and they bite into the husk, and then they strip out the tender in their inner portion, and that's how they acquire the infection. The encysted metasiccaria, which is on the outside of the husk, actually now gets into the oral cavity of the person. They swallow, of course, and then this goes to the small intestine and hatches, becomes an immature adult, and then grows up and eventually produces all these signs and symptoms that we've just heard about.
I'm very familiar with this because you do a lecture on it.
Yeah. Yeah. Well, that's. I didn't forget what I said.
I've listened many times.
Yeah, you were awake for the most part.
Well, I listened when it was recorded, and then
that's right.
As I was editing it.
Yeah. So water caltrop, as I recall, water caltrop is one of those water chestnut-like plants that have commercial value in Southeast Asia. And the water caltrop itself is not infectious because the husk is gone already, right? They don't sell the husk. And that's how they get rid of the husk. It's not a mechanical thing. It's done by humans.
Daniel, you want to weigh in on something?
No, I was just going to throw in the clinical presentation.
Oh,
so
please.
a couple things.
And now for a clinical vignette.
with more details from Dr. Rutton.
Exactly. So eosinophilia is fairly typical, even in light infections. So we had that in our case, and the man feeling swollen. There's often an allergic response, right? We're seeing this eosinophilia. So people often feel just diffusely swollen, and this is like an allergic type reaction. So it could be the face, it could be throughout. So you see that. Fortunately, is that usually these, if untreated, they're apparently self-limited about a year. But in a lot of these areas, as some of our listeners wrote in, there could be repeated infections in certain areas. It's estimated 10% of the population just chronically, repeatedly infected.
Right.
Yeah,
and there are reservoir hosts for these also, as I might add. So, particularly for Fasciola hepatica, it's called the sheep liver fluke, by the way. So it's more commonly found in sheep than it is in people. But I think cats and other animals can acquire Fasciolopsis busci as well.
Yeah.
So they are inhabitants of a human and habitats in these areas of commercial farming for, I guess you would call it a form of aquaculture.
Aquaculture.
Yeah, not fish in this case, edible plants.
All right. We will come back soon with another clinical case. But
stump the stars.
But until then, we are going to tell you about some science.
Yes.
Some basic research.
Absolutely.
And we have a paper selected by none other than Dickson Despommier.
I did pick this one.
Published in Science, it is called Transferrin Receptor 1 is a reticulocyte-specific receptor for Plasmodium vivax. The first author, many authors, Jakub Grushik, and the last author, Wai-Hung Tam. And they're from a whole host of different places. The Walter and Eliza Hall Institute, which Dickson knows well.
I do.
The Harvard School of Public Health, University of Melbourne, University of Singapore, Ulm University, the Cambridge Institute for Medical Research, Wellcome Trust Sanger Institute, Li Ka-shing Center for Health Information and Discovery, which is in Oxford, La Trobe University, it's also Australia, Melbourne,
Yep.
Harvard Medical School, Mahidol University in Thailand, home of Bushki, University of Sao Paulo, and the University of Otago.
Right.
New Zealand, which we were just talking about.
We were.
I asked Dickson where he would like to live, if any place, and he was thinking about New Zealand.
Well, you know me.
Have you been there, Daniel?
I have not. I would love to go, though.
Oh, you'd
I
love
hear
it.
it's spectacular.
It truly is.
Daniel, do you fish?
I do. Dickson and I are fishing buddies, so to speak.
Have you fished together?
Not yet, but we're going to do this. We're definitely going to do this.
No, Dickson, you and I have fished together, remember?
When did we do that?
Remember you invited me to your club there?
And you went.
And there was that pool, and you're like, don't go over there. You're going to hurt yourself. I thought you were like my dad or something. I'm like, I'll be okay.
No.
I knew a 70-year-old man. He fell off that rock. I'm like, yeah, I'm a little younger than 70. I should be okay.
Well, but it could still happen.
Now, this paper is about Plasmodium vivax.
Yes.
And there are hundreds of Plasmodium species, right? Only Falciparum, vivax, ovale, curtizi, ovale, wellicheri, malaria, and nolzi infect humans.
Correct.
Is that accurate, Dickson?
It's very accurate.
You're the man.
No, I'm not the man for this, but I do know that is true.
You are the parasitologist of the world.
In this room.
You're Earth's parasitology professor.
That's so nice of you to say that, but I can't accept that title. I'm sorry.
According to this paper, P. vivax is the most widely distributed human malaria parasite.
This is true.
Where would you find it?
All over the world, within the tropics and subtropics.
What's the vector?
It's the Anopheline mosquito.
Anopheline.
Always. The malaria parasites are always transmitted by Anopheline, female Anopheline mosquitoes.
They say here it has a strict preference for invading reticulocytes.
This is all true.
As opposed to red blood cells.
Well...
Yeah, I was gonna jump in and make a few comments
Go
here.
ahead.
So I read the first sentence. I was like, I already like this, and I need to rewrite Parasitic Sixth's edition because as you see here in the they have now taken ovale, right? We used to say, okay, there's 5, human malaria. We said, Falciparum, vivax, ovale, malariae, and nolzi. But now, as you see, they've realized that the ovale really splits pretty well into two subgroups.
Right.
So there's really 6 that commonly infect humans.
Yeah.
And then they start, this is a great introduction, really a lot in here. And then they talk about something that is really critical clinically, is that part of the potential severity of malaria infection, of Plasmodium infection, is which type of red cells are vulnerable to invasion, right? So I remember this sort of backwards. I remember malaria only infects very old reticulocytes, or old red cells, not reticulocytes, while ovale and the different types of ovale and vivax, they'll go after the reticulocytes, or the brand new red cells. And then the problem with falciparum and knowlesi is they can infect any red cells. So you can end up, instead of just a 1% or so parasitemia that you'll see with ovale or vivax or malaria, you could see 10% or more with falciparum and knowlesi. Or Knowlesi. Have we decided how to
Knowles, I believe it's
Knowlesi. I'm going to just keep swapping back.
Good.
No, that's
Knowlesi.
okay. It's okay. It's
Well,
fine.
this paper is all about identifying a receptor on reticulocytes that serves as the entry receptor for vivax. And so we know that this vivax preferentially invades reticulocytes, but it preferentially invades those that have high levels of transferrin receptor 1,
Right.
which is a receptor that binds transferrin, which is a protein, which then in turn binds iron to get iron into cells, right?
Indeed.
In the battle for iron.
The battle for iron. Iron Man chef.
Iron Man with the thing on his chest.
Iron Man.
Iron Man.
Right.
That could be a title for this
Could be.
episode, Iron Man.
Could be.
Now, it turns out that among the red blood cells in your circulatory system, Dr. Harvey,
Right.
TFR1, that's transferrin receptor 1, is only on reticulocytes. And as they mature into erythrocytes, TFR1 is lost.
How about that?
Now, could that be a determinant of P. vivax invasion? Maybe there's no receptor. So they already got a big clue here
Yes.
that it might be TF1. This is a transmembrane protein. And it also happens to be a receptor for some viruses.
That's remarkable.
Why is it remarkable?
Well, do viruses infect red cells?
No, it's on other cells as well. You need
that's
to get
right.
iron
Oh,
into many cells, right?
Oh, you do.
So it, in fact, it's the receptor for New World hemorrhagic fever arenaviruses, including Machupo, Junin, Guanarito, and Sabia viruses.
I'm impressed.
And it's also a receptor for a retrovirus, mouse mammary tumor virus.
I'll be darned.
It's been studied extensively. And what is cool, let me take a little diversion into viruses.
Please.
The sites on the transferrin receptor that bind these two viruses are distinct from each other, and they're distinct from the place where transferrin binds. And if you look at transferrin receptors from multiple species, you can see that there's been an arms race between the receptor and viruses. So, for example, the virus binds the transferrin receptor and gets in, and you select for hosts that may have a mutation in the gene encoding the receptor, which now prevents virus binding. But, of course, that mutation cannot prevent transferrin binding because that's an essential function.
Exactly.
So now you have a separate place where the virus is attaching. And guess what? The virus evolves to be able to overcome the mutation and get in. You go back and forth. That's what we call an arms race.
Yep.
This is very cool.
Yep.
So there is a protein on late stage P. vivax, which is the stage that invades reticulocytes. Is that right, Dickson?
I would say.
Late stage. It is called PVRBP2B. To be or not to be?
Indeed. That is the question.
And recombinant protein, let's give it a name, PV. Let's just call it PV. Yeah. Recombinant PV will bind to reticulocytes that produce T transferrin receptor 1. How about that?
That's
and
important.
if you treat reticulocytes with trypsin or chymotrypsin, that removes transferrin receptor,
Right.
and you can't bind PV to those cells any longer. It's a cool little trick, right?
Right.
That's an old Dickson trick where you would treat cells with a protease. And yeah, I think that your kind of experiment, isn't it?
I digested the entire host when I got Racaniello. I didn't stop at the membranes.
So they're getting some clues here that binding of this PV goes with transferrin receptor surface expression.
There's an interesting thought that occurred to me as I read this paper too. And the reason why I got excited about it was that, well, when do you have reticulocytes, right? I mean, you're always making them, of course. And what percentage, Daniel, if you look at a normal human being that's not in a tropical region with no extenuating circumstances like gut infections of various sorts, what's a normal count for reticulocytes?
I'm going to quote myself from earlier last week or later last week when I said to one of the pharmacy students, I said, for some reason, everybody knows that the lifespan of any erythrocyte is 120 days, meaning about 1% are reticulocytes.
1%.
But who knows the lifespan of a T cell, which is what really matters.
Oh, you finished on that note?
Yes. It's 8 hours.
How long?
Well,
then another question arises. How long does a reticulocyte stay a reticulocyte in the normal progress towards a mature red cell?
It's really a one, if you do the math, you're really talking about a 1 day process, right?
A 1 day process. So can you name another infection besides malaria that induces reticulocytosis?
A lot actually induce anytime you have blood loss, anytime, you need to produce more red cells, you're going to increase that percentage.
Think of how common hookworm is. And think of how common anemia associated with hookworm is. And then you probably can also predict that you would find a lot of Plasmodium vivax infection in those centers of endemicity. And I think that actually is true, by the way. That's why it's so globally found. It's found virtually in all tropical environments, but in more places than you'd expect it to be. But maybe it's because the people living in those areas have high levels of reticulocytes, and therefore they're more susceptible to higher rates of infection. Because when you look at the effect of malaria on the manufacturing of blood, basically, the malaria creates an anemia also. So is P. vivax actually creating its own cell type by inducing the anemia through infecting reticulocytes? And it's kind of a circle thinking.
Yeah, I think this is, when I give our 1%, right, we're talking about...
It's a lot of cells, though. I mean,
we're talking about people who do not have anemia, right? We're talking about people that have a normal amount of red cells. Now, if you're iron deficient... you get into a little bit of trouble. You can't make reticulocytes. Or if you hemorrhage, if you're losing blood, then if you can maintain enough iron in your system, right, keep enough iron in diet, your reticulocyte count goes up. It's actually a corrected reticulocyte count. So it might be 2 or 3%, right? Suddenly you drop down to a hematocrit of 30% as opposed to 40. But if you drop down, but you don't have the iron stores, you can't make the reticulocytes.
And therefore, the Plasmodium vivax goes down then. It should, at least.
Yeah. Well, I think we've run into problems, right, in places where we say, oh, these poor kids, they're all iron deficient. We got to give them iron.
Oh, yeah, I remember what happened then.
That could become a problem, right?
That's
and
true.
I think that was that sort of great tragedy where if you take people who are chronically iron deficient, chronically have a low amount of reticulocytes, and now you give them all this iron supplementation, they crank out all these reticulocytes, which are vulnerable to infection. Suddenly 2, 3, 5% of cells are vulnerable for parasitism.
Right.
So,
but people don't usually die from vivax infection.
So that's an interesting, you are right. And I think that I'll say yes, but then also going to throw in the caveat. We think of falciparum having the highest associated mortality, the highest number of people die from it. But when you look at cases in the U.S., actually, non-falciparum actually make up a significant number of deaths as well.
Okay.
So I think that's important because people I think got in the habit of writing it off. Oh, but no, it actually, there is a significant, we're talking thousands, right? Because it's...
And it's a relapsing form of malaria too. So we should add that it doesn't just go away once you treat, unless you treat the liver stages as well.
Yeah, and that is a wonderful, wonderful, like fascinating, unique thing that this Plasmodium subspecies has developed. Is it actually can have a dormant stage, the hypnozoite,
the really hypnozoite,
the hypnozoite, and that could years, right? People could be going along, everything's good, and all of a sudden, and I've seen several cases of that where people, it's been 2.5 years,
right?
And now they come in with fever and they've got malaria, and like I haven't left the country in 2.5 years. The last person we presented one, I think. I don't know if we presented on the show, but I actually have a friend I'm going to be watching the Super Bowl with, and he did some aid work in Central America, and he a year and a half later ended up in the hospital, fevers, chills, parasitemic, and I had a businessman from India recently. So yeah,
I'm not going to watch the Super Bowl. I'm going to work on parasitic diseases videos.
You can do both. You can watch one out of each.
My video station is in the basement in my office.
Oh, I see.
My wife will be in front of the fire watching. I may go for the last 5 minutes. It's probably the best time.
That's when Brady usually pulls it out of the fire anyway. So
they provide further evidence that this transferrin receptor binds PV. They have a panel of monoclonal antibodies that are directed against the transferrin receptor.
Right.
So they mix them singly with the transferrin receptor producing cells and reticulocytes, and then they add purified protein. They see if they bind, and some of them do block binding. So further evidence. And the other thing that's cool, one of these monoclonals also blocks one of these viruses, one of these New World arenaviruses, which suggests that they have an overlapping binding site, right? Isn't that cool?
That is pretty cool.
Yeah. What's the matter?
No, I want to go out and get the new edition of our book because
I'll get it. It's right here.
I need to read a hero later in the show.
No,
because I forgot that.
What's wrong with that one I gave you? It doesn't have it in it.
That's right.
Really? You added the heroes later?
We did.
Who wrote the heroes?
I did.
Well, the first printing is signed by you guys. That's why I still have it.
That's true. We'll sign the second one for you too.
Then they provide evidence that a subfragment of PV binds with the transferrin receptor. They did immunoprecipitation using recombinant proteins. And by the way, the binding of transferrin receptor to PV does not require transferrin. Okay, so you don't need transferrin around.
All right.
So, but I'm sure it's always there in the blood, right? So it doesn't matter. But that's interesting. They do some fluorescence assays also to show interaction between PV and the transferrin receptor. That's great. And then they find that so the virus, these hemorrhagic fever viruses, they're envelope viruses with glycoproteins in their envelopes, and they show that the viral glycoprotein binds to the apical domain of the transferrin receptor. That's known from before. And they want to know if PV competes with binding, and they make soluble viral glycoprotein, and they show it competes with PV for binding to the transferrin receptor. So they're binding overlapping competing epitope. That's pretty cool.
Very cool.
I thought that was interesting because the two different kinds of viruses that bind transferrin receptor, these hemorrhagic New World arenaviruses and the retrovirus, they bind completely different sites. They don't have overlapping binding sites. So it's interesting that this Plasmodium protein is overlapping with one of the viruses. Anyway, they did the crystal structure of this PV protein from vivax.
Not an easy thing to do.
Not an easy thing to do. It has a rod-like shape, a boomerang-like shape.
In Australia, did you say?
Yeah, shrine. In Australia, a lot of stuff.
Results kept coming back to them again.
Coming back to them. Now, what's interesting here, they look at sequences of these proteins from different isolates, and there is a lot of variation in certain positions of PV protein. So they say there probably is balancing variation between the PV and the receptor. So, as I said, for the virus, one changes and the other changes back and forth. So that's pretty cool as well. And in fact, this N-terminal domain, they have a subfragment, and that binds erythrocytes as well. So it's a very specific part. Now, here's a cool part that I really liked. I'm going to skip some of these intermediate physical measurements, which I don't think tell us very much. They wanted to knock out the gene encoding the transferrin receptor in cells. Okay, they used CRISPR to try and knock it out in an erythroleukemia cell line. All right. They couldn't knock it out. They couldn't get any cells that lack the gene, but they did get a cell line that has a deletion of a glycine at position 217 in the apical domain, which is part of the important domain for binding of PV. And they say, this if you try and delete the transferrin receptor gene in mice, it's embryonic lethal. Obviously, it's an important protein. They say, well, maybe that's why we couldn't get it in this cell line as well. Anyway, they make a protein lacking this glycine, which got deleted out of the cell line, and it doesn't bind PV protein at all. This single amino acid removal, glycine at position 217.
It's a pretty benign amino acid, too, isn't it?
Very benign. In the transferrin receptor, blocks totally the ability of PV to bind.
Weird.
So the clue is to make everyone transgenic for a transferrin receptor lacking glycine 217, and then they'll be resistant to infection.
Right. At least the Plasmodium vivax.
It would be interesting to look through and see if there's been any selection. Like, are there individuals?
Yeah, that would be very interesting. That would be very interesting for sure. Absolutely, that would be very cool.
And did they test the virus also to see whether they were not susceptible? Well, they couldn't do that though, could they?
Well, they have the glycoprotein. They could test to see if that binds, but they didn't do it though. That's an interesting question. Yeah. Very good. That's a very good question, Dickson. And they go back to the cell line where the glycine has been deleted from the transferrin receptor, and PV doesn't bind to them either.
Right.
So that's very interesting.
Yeah.
Now the question is, can they use antibodies to PV to block invasion in vivo? So they have...
that's a much more important question.
Well, they just use reticulocytes, actually. They don't use animals. Is there an animal model for vivax?
Oh, Aotus monkeys, maybe?
That's tough.
A spleen removed?
That's tough. Well, they just have a culture system where they put Reticulocyte culture with vivax, and they can see it going in by flow cytometry.
Yeah, right.
And they can show that you can block invasion using some of these monoclonal antibodies. Again, they're directed against the transferrin receptor.
Yeah.
They also look at human reticulocytes. And they see some difference between Thai and Brazilian invasion and its ability to be blocked by monoclonals. So they think there must be some polymorphism in these populations. And so combining multiple monoclonals kind of gets around that. They get about 50% reduction in invasion in these erythrocyte cultures. So that's the story. So you have this invasion receptor, transferrin receptor 1. Antibodies against it can block invasion.
Right.
Now, as we know, you can use monoclonal antibodies to treat virus infections. So, Daniel, does that bring up a therapeutic intervention of treating a vivax infection with monoclonal antibodies against this transferrin receptor?
I think the thing we need to make sure is that there isn't any kind of interference in the normal iron metabolism that would go with that, sort of a safety aspect. Because it is, it's an interesting, if there's a way to block it in such a way that all you're doing is interfering with the vivax binding, but you're still allowing normal metabolic processes to occur, seems reasonable. And then I guess the next step would be, can you generate that through some kind of a mimic, without having to give everyone engineered, cloned up, generated antibodies.
Right.
So, we're resource limited parts of the world we're trying to help here.
Yeah, these are very expensive, these monoclonals. You probably know you treat kids with respiratory syncytial virus with monoclonals. It's like a $25,000 course of therapy, right?
Yeah. It's amazing how much these can cost.
So a small molecule might be preferable, yeah. So is there a case for developing therapeutics? Is this, Dickson, you said it doesn't kill people typically.
Well, Daniel disagreed with me, and I think he's right, of course, because I'm not a clinician and he is. So, I'm just repeating the rhetoric that you hear about people who travel, to other countries for vacations and other reasons, and they come back with Plasmodium vivax, and they seem to survive better than the people that come back with P. falciparum. So, but at one point, P. vivax was the dominant species of malaria that people caught. Mm-hmm. And before chloroquine resistance arose, pardon me, and once that chloroquine resistance arose, then P. falciparum became the dominant species for the most cases. So I don't know, Daniel, you still haven't weighed in on this question.
No, so it's interesting. So, how do I gently word this? No, so that was historically the teaching. Historically, the teaching was that Plasmodium falciparum was really the killer. But if you look at some studies, actually a colleague of mine that we're going to go back here 20 years. There was a guy, Nick Anstey, and him and Don Granger and Maureen Hobbs. I was working with them on looking at why children in Tanzania get cerebral malaria. We're looking at the role of nitric oxide. And he actually published a paper not too long ago. He's back 2014, I think I'm going to say, where he was looking at Indonesia at the numbers of cases and the numbers of deaths from vivax. And in the center where he looked at, it was actually, they looked at like 300,000 cases of vivax infection. And you could say, oh, but the mortality was only 0.1% in that series. But you start doing the math on that and you start realizing, wow, 0.1 of 300,000 is a lot of people dying.
That's true.
So when you start looking, we start realizing, I mean, there are thousands of people who die from vivax.
Right.
Is it the 500,000 that we see from falciparum? Probably not, but there's a lot of deaths, I think, that we just previously just said, like, well, we know this is the one that kills you. We may not have had the technology to be able to identify. And so the common wisdom, I think, sort of fed on itself. You said, well, they did die. They must have had a mixed infection. Could have been from the vivax. So I think it's underappreciated how virulent vivax can be.
Gotcha.
All right. There you go.
There you go. Right.
Now, Dickson,
yes.
I know that you just looked for a hero.
I did.
But someone actually sent in a hero.
I see that.
And it's a woman.
I do see that. I knew this woman.
So I think you should read
Sure.
Hero. Read John's email as a hero.
Be glad to. So John writes, TWiP trio, greetings from a chilly and snowy Creighton University in Omaha. I'm writing to reply to Sarah's excellent request to hear about a parasitology heroine. I commend Marietta Vosges, a former president of the American Society for Parasitology. Below is the 1988 in memoriam from the University of California, which I will now read. Marietta Vosges, Microbiology and Immunology, Los Angeles, 1918 to 1984, professor. In July of 1984, the UCLA School of Medicine lost a great scholar, teacher, and friend with the passing of Dr. Marietta Vosges. She had spent almost her entire academic career on the faculty of the medical school, first in the Department of Infectious Diseases, and then the Department of Microbiology and Immunology. During her tenure, she and her colleagues developed one of the most innovative and comprehensive curricula in medical parasitology for students of medicine. The textbook that she wrote with her friend and colleague, Edward Markell, is presently in its 6th edition and is considered among the very best texts for students of medicine. Vosges served as the major professor for many graduate students and postdoctoral fellows from all points of the globe. She precepted medical students who would eventually apply their knowledge in laboratory parasitology to clinical practice, public health, and world health. She spent much of her own vacation time working in developing countries, refugee camps in Central America, and isolated mission clinics. Above all, Marietta Vosges was a great teacher. She was a creative educator who taught by example. She had scientific honesty, high standards of behavior towards colleagues and students, and she never put her name on a piece of work unless she had participated actively in the project. Her presidential address to the American Society of Parasitology, she wrote, "It is the teacher that sparks and stimulates the minds of students to produce new ideas. It is the teacher who gives impetus to students to become the most important propellant to our society and civilization, the innovator, because without innovation and originality, society becomes stagnant and decadent." a superior teacher, and is the interpreter and keeper of our past, the critic of our present, and the dreamer of things to come. End quote. Dr. Mariette Vosges was all of these and much more. And this was written by a friend of mine, actually, Larry Simpson. I didn't know Gerald Turner, and I didn't know James Seidel, but I did know Larry very well. So
you knew Marietta?
I did. Yes, she was a shining figure in most of the meetings that I went to for the American Society for Parasitologists.
Which parasite did she work
Short on tapeworms.
Tapeworms.
Yep.
Cool. Well, that's a good. We have a heroine.
We do.
That's a good, I said. All right, you can do your hero next time, okay?
Oh, she falls into that category. We'll have to put her picture in the book sometime.
Heroine. You should put her in the book. It's not there.
Why not?
Next edition, Daniel.
I think that's an excellent idea. It did slightly bother me when we went through and I realized that the sort of list of heroes all were men. So this is great. Right. I don't want to pick her because she's a woman, but we think we should pick her.
Don't have to. You can pick her because she was a great parasitologist.
Because she was a great parasitologist.
That's right.
Yep.
So
now we are at the time when we get a new case study from Dr. Griffin.
I'm on the edge of my chair.
I can't wait.
Me either.
I can't wait.
I was really torn. I spent a lot of time going back and forth about what to do, what not to do. And so I finally came to a selection.
Excellent.
Okay. Because what I had been doing, and then I had been making sure we picked a case from each of the chapters of our textbook. And we had come to the last chapter, which was actually a chapter that is in the arthropod section, right? And then I just realized that, we were getting to where people are going to say, that's not parasitism. That's, that's a guy who fell on some poisonous caterpillar and now his arm is swollen. And, I thought it was a fascinating case when I saw it, but I was like, you know what? This is This Week in Parasitism. So I regrouped. And I was surprised that I'd never presented this case. And then, of course, I had a case this morning that really got me thinking. So here we go. Are we ready?
Mm-hmm. We are ready.
This is a 27-year-old lawyer who comes in to be seen. He comes here in the New York area to be seen. He has an ulcer on his right foot. It's pretty big, right? It's 4 centimeters, so it's almost 2 inches in diameter. This ulcer is painless. It has raised borders. There's minimal surrounding erythema that he's describing. There's a little sort of red around the edge, and there's a border to it. It's not healing, and he's concerned because this has been going on for quite a while. So we say, well, what do you mean by quite a while? Tell us a little bit more. So he reports that initially there was a papule, sort of a small bump. This enlarged, then it ulcerated. So he's had an ulcer there. And then the ulcer then enlarged, and it's been more than 4 weeks. And it's just sitting there. It doesn't hurt. It's just this non-healing hole in his foot. We get a little more history. And no past medical problems. He did have some knee surgery when he was a kid. He said he was allergic to penicillin, but on a little further questioning, it upset his stomach. So I discounted that. His mother had a history of diabetes. He doesn't currently take any medications. His social history. Dickson always likes me to discuss the toxic habits. So his first toxic habit, social drinking. Second toxic habit, occupation as a lawyer.
For lawyers, that's not a toxic habit.
Oh, that isn't. I'm so confused. He's got his own apartment in the city, but then we get to our travel geographic history. Have you had any exposures? And the patient I saw this morning, I was like, you got to give me an exposure here because it won't make sense unless you do. But this gentleman, he cooperated. He said that about a month before he had noticed anything, he had been down whitewater rafting in Costa Rica. And when he came back from Costa Rica was when he noticed that, hey, there was this little bump. we asked a little bit more, and he said, yeah, I wore my Tevas, my sandals. I did get lots of insect bites while rafting, mainly when you're launching or landing the raft. He himself was afebrile, normal blood pressure, actually had a low heart rate in the 50s, pretty healthy, athletic, fit young man. And so that's actually in the physical exam. This is a normal, healthy, athletic young man, and he's doing well except for this 4-centimeter ulcer. It's on his right foot. It is non-tender on inspection. The base of the lesion is red. It's not bleeding, though. It has actually a little bit of a white fibrous coating. Around the edge, the borders are a little raised but not undermined. I feel like I'm reading my description of the case I saw this morning because I made a point of saying these are sort of critical issues that I felt. So the border is raised. It is hard. And actually, there's a width to this that goes around. It's not undermined, though, so it comes right up to the edge. And there's no significant surrounding swelling. And as mentioned, there's no eschar over this wound. It's actually this opened, non-healing wound. Now, have I given enough?
Say no more.
Oh,
say
yeah.
no more.
Oh, yeah. That's a good one. Dickson is already...
This time I'm confident.
Dickson is there.
I don't need to see the parasites.
It's a good one.
It is a good one.
People will get this because if they've been listening.
I hope so.
Or they could look in the book.
They could.
Which is all good stuff.
Absolutely. Anything to get them to read.
Do a few more emails.
Cool.
First one we did, John. We'll do Neeraj. Dear educators, I hope you are all having a wondering 2018 so far. I look forward to listening to many more hours of incredibly educational podcasting from your esteemed team. At my end, I would like to personally thank you for your insatiable desire to learn and educate the broader audience. Personally, I feel incredibly fortunate to have been introduced to the TWiCS series, especially TWiP, since the last 2 years, and have greatly benefited from listening to it. I work for a vaccine development company named Sutrovax, where we use cell-free protein synthesis to express and purify hard-to-make vaccine targets. Incidentally, after joining the company in 2015, I started working on a malarial protein, namely PFS25, which is a cell surface antigen expressed on malaria during the course of its life cycle in the mosquito and not in the human host. In addition to this protein antigen alone, the malarial GPI linker has also been shown to be a strong activator of host immune response. Thus, using our proprietary cell-free protein synthesis technology, not only were we able to robustly express copious amounts of PFS25, historically hard to do, but we could also design a non-natural amino acid site at the C-terminus to conjugate it to a synthetic version of the glycan core of malaria GPI using click chemistry. Finally, and most importantly, using this conjugate vaccine target, we immunized mice and showed that the antisera generated were broadly neutralizing and conferred protection against development of malarial oocysts in the mosquito gut. This to us was quite intriguing, and I'm happy to update that only recently this work was accepted for publication.
Congratulations.
On a different note, I'm sure at this point you must be wondering, well, that's interesting, but what's the rationale for inundating us with all these details? Well, the point of that detailed outline was to update you with the referenced research work. And thank you for your podcast, especially some of the earlier ones on malaria, since it was greatly beneficial for me when I was getting started on the project. Your educational podcast helped put me things into context and see the merit of the research we were undertaking. Even though you all weren't directly a part of the actual research, in spirit you were, none of the work would have been possible without the help of the incredible TWiP team, and in particular, the amazing storyteller that is Dr. Despommier. At the time we initiated this investigation, I had no prior research background in malaria and was desperately looking for resources to help educate and familiarize with this enormous field of investigation. In this regard, several TWiPs came to my rescue and helped me get abreast with why it's so tricky to treat malaria and what are the major initiatives that have been undertaken but unfortunately have not yielded in development of viable treatment options. Artemisinin-based combination therapies are still the primary mode of treatment, but post-infection, furthermore, even the proposed use of bed nets along with vaccination using the GSK-developed RTSS vaccine has largely been unsuccessful in a field setting to provide significant and sustained immunity. Thus, even with all the years of toil and desperate effort, we are far from controlling, let alone curing, this enormous global health epidemic. So to think of ways to help produce meaningful vaccine candidates, we utilized our proprietary platform for generating a synthetic conjugate vaccine that combined a malarial protein and carbohydrate into one entity. And I'm happy to report that the work was recently accepted for publication. Thus, as a token of deep gratitude and immense appreciation for your scientific generosity, I'm attaching a copy of the manuscript for your consideration. I hope you enjoy reading it as much as I have enjoyed working on this exciting project. Historically, the premise of vaccine development against malaria has been to use antigens that are expressed in the human host. However, unfortunately, none of those endeavors have yielded a potent vaccine candidate. To circumvent this, we have attacked the problem through designing transmission-blocking vaccines, which block the development of malaria in the definitive host. In support of this hypothesis, the published results have given us confidence that this line of reasoning can probably help put them. But then again, these are results generated in a lab setting, and whether these will pan out, who knows? Thus, overall, and I apologize if my email has sounded too convoluted and information dense, I would like to thank you and commend each one for your tireless effort in putting out these podcasts for the general audience. I hope my small token of appreciation will help consolidate and reinforce the belief that at times you might not hear back from everyone that listens and feel that we are not benefiting from these podcasts, but rest assured we are. If anything, my only grimace is that I wish I had found this series earlier. But nonetheless, I'm eternally thankful to my boss, Dr. Jeff Fairman, who has been a long-time dedicated listener of the TWiP series as well, for introducing me to this amazing world of insights into the parasitic world. Thus, please continue to march on and educate us as you have done for several years now. I know I have singled out Dr. Despommier as the most important cog in the wheel, but I am sure he will agree that without the diligent effort and expertise of Dr. Griffin and Dr. Racaniello, this journey would probably not have been as rewarding and memorable as it has been.
I thank all the smaller people that helped me get to my position here.
And if you kind folks ever visit the Bay Area, I would love to have an opportunity to meet and engage with you personally. As a starter, you're welcome to our company-wide happy hour on Friday afternoons, where spirited, pun intended, discussions happen. A listener in constant debt of your teaching best Niraj.
Wow. That's a wonderful
lovely
letter.
I will pass on to you two gentlemen the paper. Okay, would you like to have it?
Absolutely.
You can read it.
You bet.
Sounds great.
Daniel, can you take the next one?
Okay. So Dickson, is it okay if one of your minions reads a little bit here? No, I'm just kidding.
You have my blessings, my son.
Thank you. So Andrea writes, greetings, TWIPitos. Have you seen this one? And she gives us a link to rawstory.com. And I actually had read this story. It's about a California sushi lover pulls 5-foot tapeworm out of his own body. And then her comment is, first the patient extracting the guinea worm from his own body, and now this, still raining in Seattle.
Good emojis that she sends along.
Yeah, and there's a few emojis there.
That's right. John writes.
Wait a minute. But let me ask Daniel. So what is this? Was this a guinea worm?
No. It was a tapeworm, actually, right?
Yes.
So where did he pull it out of his rectum?
Yeah.
There's a picture of it. And he blames it on sushi. So
yeah.
What is it's anisakiasis?
No. It's probably Diphilobopterium latum fish tapeworm.
As in this article, they call it anisakiasis,
Well, that's not 5 foot long, I can tell you that.
let's take a look here and see what they say.
There's a BMJ article on this.
Oh, so of course the picture is of a tapeworm, but I don't think that's a fish tapeworm. Actually, it's interesting. You look at the picture and they are broad.
Wait.
Did you click on it here, Dickson? And the actual,
I'm going to do it right now.
Oh, and then
if the worm is broader, yeah, I see it. No, wait a minute. That's stock photograph. That's a stock
photo. But then go down. He's got this 5-foot tapeworm wrapped around like toilet paper or something.
A paper cylinder, yeah.
Wait a minute.
Looks like toilet paper cylinder.
It's still loading. So
he was sitting on a toilet when it wriggled out of him.
Well, that makes sense. That anisakiasis doesn't do that. It stops in the stomach someplace and causes great pain.
So that was actually something we talked about in the case we presented of the anisakiasis, is that it's not a tapeworm, right? It's not going to have these little...
No, it's a nematode.
It's a nematode, and it's going to try to actually invade through the stomach, right? It's confused, doesn't know what it's doing.
That's right.
And that was the Japanese girl out in Colorado. She actually went to the
that's right. Remember that
upper endoscopy, and they pulled the thing out.
They call it. There's an NPR article from the next email. They call it the Japanese tapeworm.
Japanese tapeworm.
So is that correct? They say it's a tapeworm, and it.
You know what? I think it's probably the same one that we talked about, right? It's probably the Diphilobopterium.
Yeah, it's Diphilobopterium. That's right.
It's Diphyllobothrium.
And then it's how do we pronounce that? Nicocaensis.
Correct.
Oh, right. That's right.
You see the central dark area in each of those proglottids. Those are eggs.
I see.
And that's where the pore comes out.
Wild
and
salmon.
Also, if you look at the one on the far right, you see the narrow banding of the tapeworm. That's definitely a fish tapeworm.
So he ate fish that wasn't frozen, probably, right?
Yeah.
I think similar to the man that this is very timely, I think, getting sent in. So we had that gentleman who was up in the Northwest, and the salmon had not been properly liquid nitrogenized.
Liquid nitrogenized, that's right. That's right. Solidified.
Inventing words.
Appropriately this time.
But
Oh, I see.
So it is. Yeah. So it is a tapeworm as opposed to. But then I think people get confused. So
it's not anisakiasis, that's for sure.
So
no.
Andrea was referring to a different story, I guess.
Perhaps.
A patient extracting a guinea worm was a separate story, and now one. Now this one. I got it.
That's right. That's right.
All right. Can you take the next one, Dickson? Which is the same story.
Anthony.
Read it. John
writes, TWIP trio, greetings from the snowy Omaha, Nebraska. Creighton University's snowy day gave me some time to listen to your latest episode. Oddly, this made the news right after you posted TWIP 145 and the case study featuring Diphyllobothrium. Gives a reference, same reference almost. The identical reference. The man with the tapeworm complained of bloody diarrhea, but most sources I've consulted, including the 6th edition of Parasitic Diseases, states infection with the fish tapeworm results in no obvious symptoms. Infections with multiple worms may cause nonspecific symptoms such as watery diarrhea, fatigue, and rarely mechanical obstruction of the small bowel. Page 359, by the way. Question. Did the man's tapeworm infection result in the bloody diarrhea, or does he have something else to worry about? He has something else to worry about. I can get confirmation from my physician
I will confirm. He's got something else going on.
Thank you so much. So, I think that he's probably got 3 or 4 things going on at the same time, but for sure he's got to get rid of this tapeworm, and praziquantel will be the drug of choice for that, I'm sure.
All right, one more from Anthony, who sends a link to a letter in an article in the Manchester Evening News about a toddler who fell in the park playground, wiped dog poo in her eye, and there's concern that she's going to have toxicaricanus. So Anthony wants to know, I'm sending it. I have 2 questions. Could the roundworms directly infect the eye by fecal contamination? And Dickson is shaking his head. No. Even if that is so, could the infection progress to such an extent in less than 24 hours? No.
And here's the reason. The egg itself contains a larva, if it's an embryonated egg. If it's in feces, fresh feces, it's not embryonated. So this worm takes about 2 or 3 weeks, just like the other ascarids for humans. It takes 3 or 4 weeks to embryonate in the soil. So let's assume for the moment it's an embryonated egg and it's got a larva inside. The larva won't hatch until it receives the right environmental cue. The right environmental cue is received when the worm first passes through the stomach in an acid environment and then into the small intestine into a basic environment. This induces the synthesis of an enzyme which allows the worm then to penetrate out of the egg through the small operculum, which is not very visible in this egg, and then penetrate the small intestine and get into the blood supply. So this could not have happened in the eye because that's an aberrant site. And it doesn't give it any clue whatsoever as to where you are.
Hey, Daniel, in this article, doctors say they may have to remove the eye.
That's for other reasons.
I read that. I was a little bit frightened here. I agree with Dickson. This does not make biological sense. I hate to criticize the Manchester Evening News.
Yeah, right.
But what I think she probably actually ended up developing probably a bacterial.
That's correct.
Periorbital.
You got it.
Prefrontal
it would be
cellulitis. And,
horrible.
I don't think it had to do with any kind of. It wasn't R worms. Can't blame R worms.
Nope.
It just biologically doesn't make sense. So I'm glad that nobody went and took out this poor child's eye.
Exactly.
So the mother says she has scratches on her cornea and will be 75% blind. But
she could have a corneal transplant.
But that might have happened from her falling and scraping
that's it. Correct. That's correct. So that's a fixable thing. You can actually have a cornea transplant and be perfectly normal afterwards.
Yeah.
I'm trying to figure out how they confirm. They say hospital tests confirm toxocariasis. I mean, they probably did serologies, and she probably is, serologically positive. A lot of kids, I think, if you look at some of Peter Hotez's work, we underestimate how many kids at some point got toxocariasis that's infection. Right. But I don't think this episode was that. I think this episode, they call it cellulosis, but I think it was cellulitis. Yeah. So it was a periorbital cellulitis, which is a potentially deadly bacterial infection. Hopefully you treat someone like this with high dose IV antibiotics, they get better. But
yeah, they did treat her with antibiotics, they said. So
yeah,
I presume that's what it was.
Right.
Confusing agents, huh?
Yep.
All right, we have a few more. We'll save them for next time.
Yep.
Because we have held your ears long enough. This is TWIP 146. You can find it at microbe.tv/TWIP, where you have all the show notes and the case study if you want to read it, references and so forth. Or if you just want to listen, you can go to your favorite podcatcher on your phone or your tablet and just search for TWIP This Week in Parasitism and subscribe. Please subscribe so that you'll get every episode. They're just 2 a month. It's not so bad. And if you'd like to support us financially, we'd love to have your support. You can go to microbe.tv/contribute. There are a number of ways you can do that, and we would be eternally grateful for your help. Questions, comments, case guesses, TWIP at microbe.tv. Daniel Griffin is at Columbia University Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Hello. A pleasure as always. Goodbye.
Dickson Despommier is at Racaniello.org and thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
Pleasure as usual.
You like this episode?
Loved it.
I'm Vincent Racaniello. You can find me at virology.ws. The music on TWIP is by Ronald Jenkins. RonaldJenkins.com. Thanks to ASM for supporting TWIP. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWIP
is parasitic.